Good afternoon. I'd like to start by thanking the organizers for the opportunity to talk to you today about the company that I work for called SironRX Therapeutics. The company was formed in late 2010 to leverage proprietary technology that was originally developed at the Cleveland Clinic, focused on stromal cell-derived factor 1. That technology was then taken by another organization, Regenerative Therapeutics, and they made a therapeutic product out of it. They've taken that therapeutic product, which is a non-viral plasmid-based gene therapy that expresses SDF1. They've taken it into the clinic in phase 1 studies and a couple of phase 2 studies focused on the cardiology space. Our focus is on taking that same therapeutic and moving into the dermal wound space. Our specific areas that we're looking at are scar mitigation as well as advanced wound healing. To date, we've been funded through a Series A of about $3.5 million, as well as about $1 million in non-dilutive state grant funding. And that funding is to be used to advance into additional preclinical models of dermal wound repair beyond those that supported our IND, as well as to get through a phase 1 study that's focused on surgical wounds. To date, there are no approved prescription products that are for scar mitigation or for accelerated wound healing. Everything that's on the market today is there by virtue of being an over-the-counter product or through the 510(k) route. We believe that there are significant challenges with the current products that are on the market, and we believe that our product has the potential to provide an answer to the current challenges. First of all, SDF1 is a naturally occurring protein that's extremely well understood from a biology perspective. The mechanism of action is clear. It works across anatomy types, across wound types, acute wounds, chronic wounds. The products that are out there today are typically specific for a specific anatomy or for a type of wound. They usually treat chronic wounds or they treat acute wounds. Our product is in the process, in our indication, specifically going through a prospective randomized double-blinded study. It's also, as I mentioned, it's been through a previous phase 1 and is in a couple of other phase 2 studies that are all of the same nature. Most of the products that are on the market don't have the same level of clinical validation that our product will be able to bring to the market. And lastly, from a delivery perspective, our product is provided at a single point in time, does not require further dosage over time. It's at that one point in time in our indication when the wound is closed. There doesn't need to be administration over weeks or over months. When we look at the market opportunity that we serve, it's large and growing at a relatively fast rate. In the U.S. alone, there are over 15 million surgical procedures per year that have some kind of incision, and therefore the opportunity to create some kind of scar. There's even a larger number of people in the United States that already have scars, and there's nothing that can be done about them. When we look at things from a wound care perspective, it's about a $15 billion market. It's growing at a relatively decent rate due to the underlying market drivers from a demographic perspective and from an access to healthcare perspective, and it is still significantly underserved. There are still large unmet medical needs that we believe products like ours can continue to address. So the product itself is called JVS100, and as I mentioned, it is a DNA plasmid therapy that encodes this protein called SDF1. SDF1 is a naturally occurring chemokine that's known to be one of the main proteins involved with tissue repair and restoration. And it functions by serving as the body's main homing signal for bone marrow-derived stem cells and other progenitor cells. So it tells the body at the site of a wound, this is a location that you need to fix, and bone marrow-derived stem cells and other early cells move to that location. The challenge is naturally SDF1 peaks at about 2 hours after a wound, and then within a couple of days it's down to baseline levels. What our therapy is trying to do is extend that therapeutic window from a couple of hours or a couple of days to a couple of weeks so that you get a longer-term natural healing process. Get proliferation of endothelial cells, reduction locally of apoptosis and localized inflammatory response, and really at the end causing growth of tissue and promotion of tissue repair. Our product platform, we believe, has broad opportunities to it. We're currently focused from a clinical perspective in a sternotomy wound study, which I'll talk about in a couple of slides in more detail, using an injectable formulation of the drug. As we look at our phase 2 studies, they'll be more focused on the cosmetic surgical space. There's also general surgical applications, all of which using the injectable formulation. We also believe that there's opportunities to look at reformulation, looking at topical deliveries, bandages, gels, and creams. And we have some preliminary data in those areas as well, but right now our main focus is on the injectable side and on the surgical side. Our confidence is high in the therapy. Between the work that we've done and the work that Juventus has done, there's been a large amount of animal safety data that has been generated. I mentioned earlier that Juventus has been through a phase 1 study with the drug. They're currently enrolling in 2 phase 2 studies, one for heart failure and one for critical limb ischemia. And then the work that we've done from a preclinical perspective is in the dermal wound side. And then we also have an ongoing human clinical study. So all in all, there's been over 100 patients to date dosed with the drug. I mentioned earlier that to date we've been funded through a Series A. It was about $3.5 million, and that funding is really to do a couple of things, to do additional preclinical work on the validation side for the drug in wound models, as well as to get through a phase 1 safety study that's focused in surgical wounds. So I'm going to talk about both of those in a little bit more detail. So when we look at a robust model for scarring, there's a porcine model called the red Duroc pig, which is known to be the gold standard model for hypertrophic scarring. So what we've done in this model is we've created a number of full-thickness excisional wounds around the dorsal midline of the animal. Each of these wounds is about 5 centimeters long, about a half a centimeter wide. It's a full-thickness excisional wound. We also tattoo the animal prior to the procedure so that when the animal heals, we have a sense of where we actually started from. Across each of these wounds, after they're closed, the injection is made of the drug or the placebo, or there's no treatment given. We also follow each of the wounds over a number of different time points using 3D imagery so that we can get to more quantitative data around how the wound is healing over time and what the impact is from a scarring perspective. So here's an example. This is day 28. This is one of the control animals, and you can see that we have 6 wounds that we were able to create on this animal. You can see a number of wounds are still open, and those that have closed remain with this significant hypertrophic scarring. Here's an example of one of the control animals, and really what you mainly see are the tattoo marks, and you can also still make out the fine line scars from the initial incision. I mentioned that we also do 3-dimensional imagery across each of these wounds, and this is a way for us to start getting more quantitative data around the impact that the drug is actually having. Here we're looking at scar volume and surface area, and you can see from across the ranges of drug that has been dosed, there is a significant reduction in either volume of scar or the surface area of scar compared to those that had no treatment or those that were just given placebo. This is also from a wound healing perspective. We also have a significantly improved rate of wound repair. So here we're looking at, once again, drug versus the control, and we're looking specifically at the % improvement of that visible defect. So we can use these 3D images to understand where there's still wound, where there's healed tissue, or where there's scar. And we do see over a shorter period of time no wound and no scar in the tissue in the wounds that were treated with drug. And this is a trend that holds throughout the full 90 days of the work here. A number of those wounds, you can see the hyperpigmentation. You can still see the significant hypertrophic scarring that remains versus in a number of the control wounds. You can really make out still the tattoo marks, and you can see the fine lines that remain. The other part of the funding that we received was to get through a phase 1 study. So we're currently enrolling in a sternotomy wound study. So a sternotomy wound is the wound that remains after an open chest procedure, after a cabbage procedure, or after a valve replacement. These wounds are typically 15 to 25 centimeters long. We chose this as a model, not that this is the ultimate commercial opportunity, but it's a very large wound. These are patients that are followed for a long period of time by their physicians. It's also an area of the body where the surgical procedures are fairly consistent across surgeons, and it's also an area that doesn't get a whole lot of movement or flexion or torsion, so it creates a wound that stays there and doesn't get a whole lot of other confounders associated with it. So this study is a randomized, double-blinded, placebo-controlled dose escalation study. We're looking to enroll 26 patients in 3 different cohorts. We're currently enrolling in the very last of those cohorts. We anticipate within the next month or so we'll be complete with enrollment. It's primarily a safety study, so our endpoint is 6 weeks, and for every patient that's gone through the 6-week endpoint, there's been no indication of any impact that the drug has had on any adverse events. From an efficacy perspective, we're looking at primarily from a 3-month perspective, although we follow all the patients out through 6 months. So the way the procedure actually works is after the wound is closed by the clinician, the sterile field is broken down, and then depending on the size of the wound, a different number of injections are provided around that margin of the wound. We actually use a needle-free injection system largely because it reduces the variability between injections, even between each operator, but certainly across different sites. So each injection we know is getting to the same exact depth at the same exact rate, and it's being delivered at the same exact geometry. We anticipate enrollment really to be completed within the next couple of weeks, but definitely by year's end. So to get to a couple of summary slides, when we look at the story, the company was founded in late 2010. In August of 2011, received its first influx of capital, and that capital is really focused on validating in additional preclinical studies as well as to get through a phase 1 study in surgical wounds. As we look out over the next couple of months, as we close out the year, what we anticipate will be completed will be the preclinical study will be completed that I mentioned. This is additional studies beyond those that supported the original IND. The current wound care study will be completely enrolled, and we will start to get interim data on that study. Juventus will have finished their 2 phase 2 studies, and when you combine that with our phase 1 study, there'll be close to 200 patients that will have been dosed with the drug across all those different clinical studies. We are currently working with the regulatory authorities on our phase 2 protocols. We've already submitted one of our protocols to the RAC, and the other protocol we've submitted to the RAC but got bounced back because they're currently all furloughed. So we anticipate hopefully in the near future that our other phase 2 protocol will also be approved, and that will allow us to go to FDA and work with FDA on the protocols. And lastly, we also anticipate additional IP on the product and on methods of using the product will also be allowed. So where do we go next? We're currently advancing into protocols that are focused on their phase 2 protocols, but are focused on cosmetic surgical wounds as the opportunity. We also believe that there's opportunities for us to continue to enhance the product platform itself, be it through enhancements to the delivery device or additional R&D work around formulation or around development of additional assays that we know we're going to need to support in the end of phase 2 and then into phase 3 studies. So from a fundraising perspective, we are starting to investigate our next round of funding, and it will probably be up to about $12.5 million to allow us to get through a couple of phase 2 studies and also to do some additional R&D work. We believe that since the initial investment that came in a little bit over 2 years ago, we've been pretty capital efficient, and we've created a significant amount of value for the organization. We also believe that there's significant opportunities for us to continue creating value for the organization and our shareholders as well. So with that, I think I have a minute or so. If there's any questions, I'd be more than happy to address them. Okay. Thank you for your time.